But what we're going to do today is we are going to finish off our skeletal muscle contraction. Now understand, this can also basically apply to cardiac muscle contraction. So what we're really talking about is striated muscle contraction using skeletal as our example. Now we had 3 stages. The first stage happened at the synapse. Do you remember what that synapse was between the nerve and the muscle?
It's like the gap.
Neuromuscular junction. That's where you find the synapse. And so that first stage is the excitation stage, where you transduce that electrical signal in the axon to a chemical signal. The second stage that we covered last time was the coupling, where we took that chemical signal and changed it back into an electrical signal that flowed across the sarcolemma of our muscle down the transverse tubules, causing the release of calcium. Now we're at the point, we talked about this kind of all throughout, especially when we were introducing the thick and thin filaments and looking at their shape and everything. So now we're at the point of getting a muscle contraction. So this third stage, some refer to it, I believe your textbook refers to it as the Crossbridge cycling. I just like to refer to it as contraction. I like one word, simple, easy. Excitation, coupling, and contraction. So we're going to finish that off here, if I can get it to work. So there are 3 stages. We are down to step 3, which is our cross-bridge cycling, aka contraction. And like we did previously, we have a video that we're going to watch.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the Crossbridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. The activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. ADP is released, and when another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Crossbridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin-binding site on actin.
A lot of steps, right? All right, like we've done in previous topics, we're going to walk through these one at a time. I wanted to give you the overview of what's going on first. And again, did you recognize some of the pieces already? Some of the things that we have talked about? So one of the first things to remember, the myosin molecule. And remember, what did we say? That it wasn't like a golf club, but it was like what? It's like your arm. And when we look at the globular heads, that's for grabbing stuff. So when we think about a contraction, you have to understand that before the contraction, your hand is already filled with ATP that has been broken down into ADP and phosphate. When you have those molecules present, it's as if you have a bow, you're going hunting. When you break down and release that energy, you pull back the bow. To shoot that, you just have to let it go, right? So that's going to, as you heard in the video, that's going to cock the myosin head just ready to form that cross bridge. But it can't because that site is blocked on the thin actin. So we've already preloaded the myosin head. Calcium comes in. And where do we store calcium when the muscles relax? What organelle? The SR, sarcoplasmic reticulum. And that's where we're going to be releasing the calcium from when the action potential comes down the T-tubule. Now, so far, we have only in muscle talked about one calcium binding molecule. Do you remember what that calcium binding molecule is on the thin filaments? We've got two T words. It's one of the two T words. Troponin. Troponin consists of 3 proteins, and there's a calcium binding part. So calcium comes in and binds to troponin. That causes troponin to change shape. Now, troponin is also hanging on to actin and tropomyosin. So when troponin changes shape, it's going to be the engine that moves tropomyosin out of the way. And when we say in the way, we're talking about the myosin binding sites on actin, the little black dots. So the calcium was released to expose the myosin binding sites on actin. Now, our myosin was already pulled back. It's already cocked, ready to go. As soon as that binding site is open and available, you form a cross bridge. This is where that myosin head snaps up. And again, this is why I like to use my arm. Think about this is the cocked position. When that myosin binding site's exposed, you just rotate your hand up and you grab the actin. That's that pivot point I like to think of with the wrist. Your hand is still filled with ADP and phosphate, but it's also able to reach up and bind that site on the thin filament, the actin molecules. Here we can see a relaxed muscle. Troponin is illustrated as the little lifesavers and the green string. That's tropomyosin. And when we have calcium, it moves that tropomyosin out of the way. Now you see we have a cross bridge form. No contraction has started yet. We've only grabbed the actin, getting ready to move it. And that's the formation of the cross bridge. Now, the way we get one contraction cycle And the trigger for doing that is going to be the release of the phosphate. So here in this illustration, we can see that we're relaxed, we're not connected, we have ADP and phosphate on the myosin head. But as soon as that gets moved out of the way, we form a cross bridge. And as soon as you form the cross bridge, the inorganic phosphate molecule is ejected, leaving only the ADP and the cross bridge present. So when the binding sites are exposed, we get cross bridge, phosphate leaves, and as soon as the phosphate leaves, now we get the power stroke. This is the beginning of the contraction itself. We've gone through a lot of steps to get to this point, haven't we? Through the nerve, through the signaling along the membrane. Now we finally get to this point where we can actually start contracting. And this is where the other arm comes into play. I'm a thick filament. My body is the M line, and I've got two myosin molecules on either side. So we've got ADP and phosphate. Sites are exposed on actin. Snap your fists up to form a cross bridge. Phosphate is ejected. Now you're already holding on to actin, and now actin gets pulled closer to the M line, and that's the power stroke. The myosin molecule and the energy from the ATP pulls those thin filaments closer to the M line. So cross bridge, phosphate leaves, power stroke, and you begin to shorten the sarcomere. Now, hopefully you appreciated from the video, and maybe you couldn't see it if you're kind of in the back, but if you watch the video on your computer or your smartphone, you will see at the end of the video as that sarcomere is contracting, those myosin heads aren't just moving once. They're moving like the pistons on your car. It's going to take several cycles from each myosin head to completely shorten the sarcomere. It's not one and done. Again, it's like the pistons. So it's going to be the cycling over and over and over to shorten the sarcomere. So, but once we're fully contracted for our myosin molecule, we've got to release and do it again. So this next part, I'm not going to say there's a little bit of controversy, but there's a lot of confusion out there, including on the video. Did you notice there was a part of the video that seemed to kind of jump and skip? That's because I edited it, because it wasn't correct. And your textbook isn't correct. So, this is how we're going to learn it. So once you get the power stroke, and there is debate on if the ADP leaves or if a new ATP molecule comes in and kicks out the ADP. Either way, I'm good with either way, but the thing that ADP has to leave to make room for the ATP molecule. So the way we're going to learn it, and the way I've taught it for the last 7 years, is after the power stroke, to release the cross bridge, ADP has to leave, and a new ATP molecule has to bind. The binding of the new ATP molecule releases the cross bridge. And then the breakdown of ATP into ADP and phosphate resets us. It pulls back the bow so we can do it again. And as long as that binding site on actin is exposed, which means as long as calcium is present on troponin, which means your nerve is continuing to signal, those myosin heads are just going to keep going through that cycle. Just keep contracting. Now, you don't feel it, jerky like this. You feel it as a constant contraction. We call that tetany, and we're going to learn about that next week. All right? So at the end of the power stroke, we're going to say ADP is released, ATP comes in. But you understand it may be that the ATP is kicking the ADP out. They either happen simultaneously or they happen in sequence. But the important thing is the ATP breaks the crossbridge, and the hydrolysis of ATP releasing that energy resets and recocks the myosin head to do another cycle. And you have to be able to do that for a complete contraction. That's our cycling. Have we got that? We're going to go through it 2 more times before we finish today just to make sure. But that's going to continue to happen until the nerve stops signaling the muscle. When the nerve stops signaling the muscle, meaning your muscle needs to relax, then those calcium release channels on the sarcoplasmic reticulum are going to close. You're not going to release any more calcium. But we've already got a lot of calcium out there, and the only way we can relax the muscle is remove the calcium. So in addition to closing the calcium release channels, we also turn on calcium active transport pumps that vacuum the calcium back into the sarcoplasmic reticulum. And as you remove the calcium from those thin filaments, then the contraction will stop and your muscle relaxes. Because on that very last power stroke, myosin's hanging on to actin. But when it releases that last power stroke, when it's reset with ATP, the tropomyosin already knew it had to slide over those spots. And when it releases that last time, it slides over, and there's nothing for the cocked myosin to bind again. And that's the relaxation.
Is the calcium originally coming from the cisternae?
So the terminal cisternae is that enlarged part of the sarcoplasmic reticulum, so yes. Yeah. And that's where it gets pumped back into. So once again, a muscle contraction, you've got to use 2 molecules of ATP for every cycle. You've got it preloaded with one, and you've got to use another to reset it. And now we also have to use ATP to remove the calcium, to relax the muscle. So there's a lot of energy involved. Do you see why if you don't use your muscle, you lose them? Your body's not going to waste energy maintaining something you're not going to use. All right. So here's a little cycle. Biologists like cycles and life cycles and all that. So we put this one together. We're starting with a preloaded myosin head. And we're saying we got our thin filament that has exposed binding sites. We've already done the calcium. We've already done the sliding of the tropomyosin. So we just exposed the binding sites. We form a crossbridge. What has to leave before we can get a power stroke? The phosphate. And when the phosphate leaves, which is the little gray guy there, now we get the power stroke. Now, at the end of the power stroke, you can see in this illustration and from this other textbook that I've used, ADP is released and a new molecule of ATP is able to bind in that exposed site now. That breaks the crossbridge. And as long as the myosin binding sites of actin are unexposed, we're going to do it again and again and again. And it's just going to keep going until you either run out of ATP or the nerve stops signaling. Now, is there ever going to be a time you're going to get a muscle contraction and you're going to run out of ATP and your muscles can't relax? Is that ever going to happen? It happens one time in your life, and you don't look forward to that day. You ever heard of rigor mortis? Rigor mortis literally means the stiffness of death. And typically at the point of death, your central nervous system has this explosion of electrical activity. You look in the westerns and some scary movies and stuff, the guy that gets killed with a gun in his hand, and they can't pry his fingers open to get the weapon. That's because he has this death grip because that explosion of electrical neural activity has caused his muscles to contract and continue to contract. And that contraction is going to happen until all the ATP is used up. Now, your heart's not pumping. You're not getting more glucose. You're not getting more oxygen. And you're eventually going to run out of ATP. And what do you need that last molecule of ATP to do? Reset. So your muscle fibers can't reset. They're just locked. But over time, your body becomes less stiff, not because you're making more ATP, but because the proteins and the cells are dying. They're breaking down. And so that's how a coroner or a medical examiner can examine a body. And if they look at and see an amount of rigor, stiffness, that gives them an idea about the time of death. If there's some rigor, I believe it's within maybe about 24 to 36 hours. If there's no rigor, it's past that point. So they go, okay, no rigor, then this body's been here at least this long, and they begin to look at other evidences. But that's where that comes from, just running out of ATP because those cycles just kept going and going and going. There was nothing to stop it. So we've gone through excitation, we've gone through coupling, and now we finished out contraction. So what I wanted to try to do you remember when I showed you the abbreviated stuff, the outline stuff? How I would try to put this together so I can remember it? That's what I'm trying to do here for you today. So when you put this whole cycle together, we're going to have 4 steps in excitation, so E4. We're going to have 5 in the middle, which is C5. And the last one's going to have 6 steps, so C6. You can do it any way you want to do it. By remembering the letters and numbers, it helps me know, okay, if I missed anything. So let's look at excitation. What do you think the A little cp dealing with a nerve, what might that be? The action potential. This is the electrical signal. And do you remember the name of this nerve? What kind of nerve signals a muscle? Motor. It's a motor nerve. So again, I didn't put M, I didn't put motor, but you're going to remember that. See, these are just some cues to get you started memorizing. This is not the extent of all you need to know. Does that make sense? The more you can memorize and not look at, the better prepared you're going to be for the exam. So the motor neuron signals via an action potential. What's happening there with number 2? What's VG? Voltage gated. Because of the flipping of our negative and positive inside membrane potential, that's going to trigger the opening of calcium channels in the membrane of the nerve. Now, I didn't put it here because this is something that you need to have memorized already. When you open the calcium channels, where does calcium go? In. It comes in because like sodium, it's high concentration out. Calcium comes in, and what's calcium going to lead to here in number 3? What's going out? Acetylcholine. What kind of molecule? If you had to characterize acetylcholine, what kind of molecule is acetylcholine? A neurotransmitter, a chemical signaler coming from the nerve. Everybody up so far? All right. But do you see how we're not writing everything, but you're doing a pretty good job keeping up already? Last, acetylcholine binds to the receptor. Where are the acetylcholine receptors? What is that? What is that? Yeah, the wavy thing. What is that called underneath the terminal knob? Motor, say it louder. Motor end plate. It's on the muscle. Acetylcholine's coming from the nerve. The receptors are on the muscle in that increased surface area folding of the motor end plate. All right? Any questions about excitation? This is just a bit of review about that point. Anything we've left out here? Can you think of anything we've left out in these steps? Anything you would like to put in here? I put this together this morning pretty quickly. Okay, we talked about the vesicles. Vesicles would be sort of 2.5. Because the acetylcholine is stored in the vesicles, what causes the vesicles to release the acetylcholine? The calcium. But again, if you want to put that information in, if you're having a hard time memorizing it at first, go ahead and put that in as an extra step. This is not, perfect for everyone. Make it fit the way you need to study and the way you need to learn this, okay? This is a starting point. And to give you an idea. So we're getting a little bit more complicated now as we get into these next two, and so that's one reason why we've added steps. So number one, when you bind acetylcholine to the receptor, what happens to the receptor? Just very simply put, when you bind acetylcholine, what does that receptor do? Opens. The receptor opens. That acetylcholine receptor is a tunnel for mostly sodium. But without acetylcholine, it's closed. So, acetylcholine opens sodium channels, and I kind of wrote this in this way. MP flips to positive. MP. Membrane potential, because remember what was it before? It was negative. But when that sodium comes in, it flips to a positive. That's a switch. It's like turning a light switch on and off. So when we flip our membrane potential from negative to positive, right next door to our neuromuscular junction, we have VG. What's VG? Voltage-gated who? Sodium channels. And when voltage-gated sodium channels reach their threshold, they open. Do you remember that number? I told you to remember this number. Negative 55 millivolts. When you reach negative 55 and sodium voltage-gated channels open, what have you just reproduced? Another action potential, this time not in the nerve, on the muscle. And that goes across the whole membrane, including, where does it go in step 3? Down the T-tubules. That's a way to increase the surface area, spread the action potential more rapidly. And we can detect that membrane potential flip from negative to positive by voltage-gated calcium channels that are linked with calcium release channels. So the action potential signals those calcium release channels to open. And when those calcium release channels open, calcium comes out of the what? Sarcoplasmic reticulum. Would you rather say terminal cisternae? Does everyone understand the relationship between terminal cisternae and sarcoplasmic reticulum? The sarcoplasmic reticulum has several different parts. The enlarged part of the SR is the terminal cisternae. It's kind of like Texas has a number of different counties. We're Nacogdoches County, we're still part of Texas. Terminal cisternae is like a county of the sarcoplasmic reticulum. Does that make sense? But ER is a little bit easier than, SR is a little bit easier than terminal. So anyway. Coupling. We good with coupling? I sort of combined a couple of things to try to keep it in as few steps as possible. But do you see why we call it coupling? Because we're coupling the chemical signal with acetylcholine with, again, an electrical action potential, and changing that signal again back into a chemical, the calcium, that's going to start the next phase, the contraction. So excitation, acetylcholine, coupling, regenerated action potential, and now the calcium is going to start the contraction. Now, I had a little bit of trouble with number 1. I almost just put an asterisk. I was like, no, we need numbered list. So you understand, we preloaded it. You can't fire an arrow unless you draw back the bow. So this is the drawing the bow. This is the cocking the gun. Myosin has to be preloaded with ATP. As soon as ATP binds, it gets broken apart into ADP and phosphate. And that causes the reset. We're ready to go. Step number 2. Calcium binding to troponin activates this troponin-tropomyosin complex, sliding tropomyosin out of the way. What is tropomyosin blocking? What is it masking?
Binding sites?
What binding sites? Myosin binding sites on actin. So we talk, we get in the habit of saying things kind of loosely and not adding all the info to it. But tropomyosin is blocking the myosin binding sites on actin. So again, we're not writing that, but that's information you need to understand. And if you need to put it in at first and then slowly get down to this little bit of information or less until finally you can recopy this stuff absolutely from memory and know it, that's when you will be ready for the test. All right? So we preloaded myosin. We've slid tropomyosin out of the way. Now, number 3, crossbridge. As soon as the crossbridge happens, what goes? Phosphate. As soon as the phosphate goes, step 4. Power stroke. As soon as the power stroke happens, what goes? ADP. But we can't let go until what comes in to break the crossbridge? A new molecule of ATP. It releases, and then we have to reset to do it again. And what do we need to reset? To break the ATP down and get that energy. And then as long as those sites are exposed, we can do it again and again and again. Any steps that we need to do here? Anybody have any questions about any of the steps? Okay. Now, before we go to this next part, do not punch your neighbor in the face, okay? You promise me. Don't go out of here after you hit somebody and say, well, Dr. Langford told me to. Do not punch your neighbor in the face, all right? When I teach this to my pre-meds, they will, one or both, be doing this during my tests, and I have been told they also do this during their medical college admissions test. All right? Remember, don't punch your neighbor in the face. Okay? Arms out. See, now you see why I said that? That's why I prefaced that. This is your myosin molecule. We're going to go through crossbridge cycling. Now remember, this is our globular head where we bound ADP and phosphate. Okay? This is our loaded, cocked, ready to go position. Now that is our crossbridge, right? We reach up. Did she hit you? I told you not to. Okay, that's our crossbridge. We reach up and grab actin. When we grab actin, phosphate has to go. When the phosphate goes, that's our power stroke. We get the power stroke, ADP and ATP come in, and we open our hand, releasing the crossbridge. And when we break down the ATP, we reset to do it again. Okay? Everybody got that? Okay, let's practice. Arms out. Crossbridge, phosphate goes, power stroke, ADP out, ATP in, reset. Okay? Not bad, not bad. Arms out. All right. Now, in our hands, we have ADP and phosphate, right? Which is the smallest, the phosphate or the ADP? The phosphate's the smallest, and that goes first, right? So, when we form our crossbridge and we have to kick out the phosphate, which is the smallest, you're going to go Don't spit on each other either, okay? Okay, you got that? Okay, let's practice. Okay. Crossbridge. Come on. Okay, one more time. Reset. Crossbridge. Power stroke. All right? Now, ADP's bigger, right? Okay, reset. I'm telling you, they do this during the medical college admissions test. I'm not kidding. They were so excited to get back. I had a muscle contraction question, and I did the... Oh. I said, did people look at you? Go, yeah, but I got it right. Okay. Crossbridge. Power stroke. Nice. Somebody got spit on that time. ADP out. Now, I don't have a sound for ATP coming in. Like that? Okay, reset. Okay, you ready? Crossbridge. Power stroke. ATP in. Reset. Oh, that's good. I like that. I like that. Have we got the cycling? Have we got the cycling? All right. Hey, that usually happens when I'm teaching, so I'm kind of used to it. All right. Now, that is our excitation, coupling, contraction, and that's our class for today. Have a good weekend. No, you're going to hear on the test, you're going to hear people going...